Bismillahirrahmanirrahim. Honorable guest speaker Dr. Manzoor Abbas, respected faculty members and dear students, Assalamu alaikum. I am Dr. Abid Hussain. On behalf of Head of Physics Department, KFUEIT University of Engineering and Information Technology, Rahim Khan, I welcome you all and thankful to all of you for sparing your precious time to participate in this webinar under title Short Peptide Conservates for Protocell Formation and Biomedical Applications. Before the formal start of the event, I would like to invite Dr. Muhammad Usman for recitation of the holy verses of the glorious Quran and Naat Rasul Maqbool sallallahu ta'ala alayhi wa sallam.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم قل هو الله أحد الله الصمد لم يلد ولم يولد ولم يكن له كفوا أحد صدق الله العظيم اللهم صل على سيدنا ومولانا محمد اللهم صل على سيدنا ومولانا محمد وعلى آله وصحبه وبارك وسلم اللهم صل على سيدنا ومولانا محمد صلى الله عليه وسلم.
Jazakallah, sir. I am presenting a brief profile of our guest speaker, Dr. Manzoor Abbas, who has earned his master's degree in the subject of chemistry from Bahauddin Zakariya University, Multan in 2010. He received his PhD degree in 2017 from University of Chinese Academy of Sciences, which is one of the leading universities in the world. He worked as a postdoc fellow at the Biological Environmental Sciences and Engineering Division, Kingdom of Saudi Arabia. In 2019, he won the prestigious Marie Curie Fellowship in Redmond University, the Netherlands, and currently he is working over there as a nanotechnology expert. Dr. Abbas has published around 16 peer-reviewed research articles in reputed journals like Advanced Materials, Journal of American Chemical Society, and Chemical Society Review. He has got several book chapters published, and his one book is in the publishing phase with the Elsevier publishers. His articles have more than 1150 citations. He keeps on presenting his work in invited talks and prestigious scientific events. He has received several international awards on research contributions and has generated research funding more than 350,000 euro. He has supervised many research students and actively participated in scientific research dissemination events for community services. Currently, he is with us to disseminate his research work with the audience of KFUEIT University, Rahim Khan. Daksa, please start your valuable talk.
Okay, thank you very much, Dr. Abid Hussain. And first, I would like to thank Dr. Saifur Rahman, Director ORIC, and Dr. Muhammad Bilal Taher, and of course, worthy Vice Chancellor, Suleiman Tahir Saib, for inviting me on this webinar. And today I am going to present my latest research, which I am doing here in Netherlands at Radboud University. First, let me check this one. Okay, it's in a. Now it is presentation mode, right? Yeah.
जी सर ठीक है यस गो अहेड प्लीज
Okay. Yeah. Today my topic is short peptide-based quacervates for the protocell formation and biomedical application. My mainly work, and in this talk I will talk about mainly for protocell formation and little about biomedical applications. First, I would like to start from this question: what is the origin of life? How life was originated? Being a Muslim, this is our belief that the universe was created by Kun and Fayakun. But still, in Holy Quran, there are many verses where Allah Almighty says that we have to investigate. We have to investigate the hidden things in the universe. Therefore, in my research, we are investigating how the life was originated. Here, there are different kinds of myths, theories, and experimental details that how life was originated. Hello, can you listen me, please?
yes sir we are listening carry on please
Okay. Yeah, thank you. And how life was originated. So recently, the many group of scientists have make a one hallmark how we can investigate the life. Firstly, we need to think about what is the cell and what is the function of living cells. So we can then better understand the origin of life. So scientists have made some parameters or hallmarks which are necessary to make an artificial cell, a primitive cell, or protocell. So first one is compartmentalization. We need to have one compartment which looks like a cell, a living cell. And other parameters or hallmarks would be those compartments should have growth and division property. And third one is information processing and energy transaction. And most importantly, they should have mobility or adaptability. So these hallmarks I am investigating here at Radboud University, and how we can go for from peptide self-assembly to make the compartments and then protocells. First, we know peptide building blocks can self-assemble to various kind of different nanostructures through non-covalent interactions, but they can also co-assemble to form one supramolecular copolymer kind of structures, which they are composite hydrogel and nanospheres, microsphere, and they all have different properties. And we can rationally design the building blocks, peptide building blocks, to make nanomaterial according to our projects. So there are several different kinds of compartments have been reported. For example, people are using lipids to make liposomes. People are using proteins to make proteinosomes, and then colloidosomes, polymersomes, and quacervates to mimic the cellular life or cell. If we simply say to make the artificial cell or the protocell. So today I am going to talk about mainly about the quacervates. Why quacervates? Quacervates have several advantages over other their counterparts. For example, it has very liquid-like properties, and its internal core is not rigid, and we can encapsulate different kinds of material inside to mimic exactly the internal entities of the living cell. So recently, quacervates have been reported as a model protocell. Quacervates itself is a new term. It's not very new term, but it was first used in 1929 by a Russian physicist and then one Dutch chemist. After 70 years of gap, the people has forgotten about the quasar weights, and they have proposed this quasar weights as a model for the, or they have thought might be quasar weights could be best option to study the living cells, or best compartments to to study the living cells. However, after 2010, it is again getting momentum to study the origin of life by using quasar weights. However, there are different kinds of quasar weights. For example, mainly we investigated complex quasar weight system and the simple quasar weight system. So complex quasar weights mean there are two different kinds of building blocks comes together through electrostatic interactions to make a compartment or a quasar weights or a liquid droplet. So one of famous scientists, Stephen Mon from Bristol University, he is using peptide and nucleotides to make the quasar weights for protocell formation. And then one Keating from USA, he is also using, she is also using RNA and peptide system to make the quasar weights. And some people also using RNA catalysis inside the quasar weights. However, to make the artificial cell or protocell, there are two main techniques. One is bottom-up approach and other is top-down. So bottom-up approach is where we use non-living components, non-living building blocks to assemble them to make artificial cell. Mean we are heading towards complexity from simplicity. Building blocks or non-living components are simple, and once they assemble together to form the quasar weights or artificial cell, they are more complex in nature. Like we know the living cell is also a complex system because there are different functions and their internal entities are different, and lot of things or reactions are happening inside the cells. And other is top-down approach, where we can take the living organisms to make some genetic coding and then take their genes and other stuff to make the artificial cell. But that is little complicated system to make a biomimetic cells. So my research is mainly from non-living components to artificial cells. It's kind of bottom-up approach. So liquid-liquid phase separation is a physical property where we use two kinds of LLPs, mean liquid-liquid phase separation. One is segregative and other is associative. So first kind of liquid-liquid phase separation or first kind of quasar weights is based on two component system. One is polymer, proteins, peptide, nucleotides. We can use two of them to make the quasar weights. And other is very new and recently, I think in couple of years, and I can say we are the first one which are introducing the simple quasarvation world and even this term for to make the quasar weights. So simple quasar weights mean we take only one building block to make the quasar weights, which can be used as a protocell model. So how quasar weights can look like? For example, they are kind of microstructure, microspheres. Many people can say these are the microstructures. Why we are seeing them as a protocell? So I can tell you why we are seeing them as a protocell model. So what kind of interactions are involved in the formation of these quasar weights? These are very common non-covalent interactions. For example, many of you know this charge-charge interactions, dipole-dipole movement, hydrogen bonding, etc. These are same interactions, non-covalent interactions, which you people have reported for the formation of different kind of nanomaterials. So what is minimal motif for phase separation. We know people are using proteins, RNA, etc. to make the quasar weights. But Martin et al. Has reported one very new technique or model, which is spacer sticker model, where they have used sticker as aromatic residues or the charged patches, mean charged residues, and spacer, which are polar in nature and flexibility in flexible region. And what is the then problem? Why we are studying for the protocell formation or the phase separation to make the protocell? So these proteins and polymers are have large size, which is more than 30 kilodalton. So it means the system is quite complex. To understand main fundamental features of a cell, we need to have very simple system which can be understand, and where we can understand its formation process, what is the mechanism of formation, and how they act as a protocell formation. So therefore, we know this diphenylalanine, FF, this is very short peptide. Only 2 amino acids are involved. And this was first published in 2003 in Science, when one professor from Tel Aviv University, he has reported for the formation of nanomaterial. But in last 2 decades, it has reported for various kind of different formation of various different kind of nanomaterials, including nanowires, nanotapes, nanovesicles, etc. Therefore, can we use this simplest system to make the protocell or the quasar weights? Yes, we are the first one who are using this FF by conjugating them with a spacer, and because we were inspired from the sticker spacer model, where we use spacer as a flexible and hydrophilic spacer or moiety, while sticker are kind of aromatic region or the hydrophobic region. Aromatic are the hydrophobic amino acid residues. Our model compound is FFSSFF, which I rationally designed during my PhD studies. So FFSSFF means it has 4 aromatic groups which are hydrophobic in nature and 1 hydrophilic spacer. So which makes this molecule overall as a hydrophilic. It means it is soluble in water. So at physiological conditions of salt and pH, which is required to investigate any kind of study which is relevant to living cells. So just we dissolve this compound in water, and the pH decreased to 6, which we didn't decrease the pH, but it was due to counter ions of that peptide. And when we increase the pH from 6 to 7.4 or so on, mean 8, 9, 10, then we observed turbidity. And this milky color, when we observed under the microscope, we comes up with these very nice quasar weights or microspheres. So these are not only microspheres, these are not only liquid in nature, but they are the dynamic in nature as well, because they can move from one place to other place and they can fuse together, which is also required hallmark for the investigation of a living cell. So I will try to play this video where you can see the fusion. For example, here you can focus on these 2 droplets, where these 2 droplets are fusing together to make one bigger, and also the other droplets are moving from one place to other. It means the droplets are dynamic in nature. So we investigated the phase diagram or phase separation properties which is required for the investigation of coacervates are the protocell. First, we check the hydrophobicity or hydrophobic nature of the FF based coacervates. For example, we monitor or titrated with one aromatic molecule which is very small in molecular weight, phenyl methyl ester. Once we added different concentration of this molecule and we observed that turbidity was just disappeared because this molecule enhanced the hydrophobic nature of that building block FFSSFF. So this is coacervates are kind of solute responsive. Next, we investigated the solvent responsive nature of the coacervates. For example, we incubated or we added titrated with acetonitrile with coacervates, and we observed after a certain concentration of acetonitrile, the coacervates were dissolved. It means there are two phases and acetonitrile can dissolve the coacervates. We made the phase diagram where we have investigated there are two phases. It's not one phase. So coacervates are the droplets or liquid droplets which can form spontaneously and separated into two regions. One is solution and other is the dense phase, which usually we called it coacervates. And we want to make this design, which is based on spacer sticker model, to make it more versatile. So what we have done, we just replace one of amino acid because FF was the model molecule. We just replace one of amino acid by tryptophan. Tryptophan is amino acid which is more hydrophobic in nature. And we replace F with the L. L means leucine, which is less hydrophobic than F. So we also replace both FF with the LL. And we observed that when we have increased this hydrophobicity, we come up with the aggregation, which is kind of random aggregation. And when we decrease the hydrophobicity, then we have still coacervates, but they are these compounds required high concentration to make the coacervates. We have also investigated the role of spacer. This first spacer is a hydrophilic in nature due to this heteroatom atom in the middle, and other we investigated C6, mean carbon 6 with 3 methyl groups, which is far hydrophobic than this molecule, this hydrophilic spacer. And we investigated and we had results that once hydrophilic spacer is there, then the coacervates were formed. And if we increase the hydrophobic nature, then we have the aggregation again, just like the WF one. We manipulated the redox chemistry for the formation of protocell, because protocell is kind of a property where we can make or break the protocell or make or break the coacervates even. For example, this model compound has disulfide bond in the middle, and we know that disulfide bond is responsive towards reducing agent. And once we added the reducing agent, that can cleave this disulfide bond, and ultimately we observed that once we have added DTT here, which is a reducing agent, we again observed a transparent solution. It means that compound was broken from the middle, and this into thiol group, and thiol group can again form the disulfide bond under oxidation conditions. So we use hydrogen peroxide and other oxidizing agents to see the turbidity again, and we observed this by microscope, the formation of coacervates again, and we have also confirmed by the LCMS, mean its formation and deformation by the redox chemistry. So we use different oxidizing and reducing agents and titrate it against to measure its turbidity and titrate it against with respect to time. And why these quasar weights are protocell? We all know living cells have genetic material inside. For example, they have DNA, RNA inside, and many other functional groups and small molecules and etc. So how we can prove that these quasar weights can mimic a protocell or can be act as a protocell to mimic living cell? So we encapsulated or sequestered different small organic molecules which are pigments or small dyes, and we also encapsulated DNA and RNA which were successfully partitioned inside the quasar weights, which again confirm the role of these quasar weights as a protocell model. And importantly, we know there are a lot of metabolic reactions are happening in living cells. That's why human bodies are alive. So how we can investigate that these quasar weights are exactly acting as a protocell model or a living cell? So we designed one very common organic reaction, which is aldol condensation reaction, by using aldehyde and ketone. Most importantly, can these quasar weights act as a microreactor like the living cell? So we have observed this by the NMR that once we use these small molecules to make this new product, which is condensation of two molecules, so we observed that there is the formation of product when we use the quasar weights, and there was no formation of product when we didn't use the quasar weights, but we use the same concentration. So this confirms that these quasar weights were acting as a microreactor or acting just like a living system or living cell. So still there are many things which can be investigated. So we use different higher concentration to see the energy barrier, where we found that when we use higher concentration, there was the 50% faster reaction rate. Yes, this was one part, but still there are a lot of hallmarks or fundamentals which should be investigated. How we can investigate? For example, I can say growth and division is not very clear. I mean, we can say these are the compartments, these are quasar weights, they can be act as a microreactor and encapsulate genetic material, etc. But can they grow to make a bigger, and can they divide after that? So recently I am working on this to investigate how the quasar weights can grow and then divide, just very similar behavior of the living cells. So we changed the rational design of same building blocks by introducing the tyrosine amino acid. And we know tyrosine is very responsive to oxidation, and especially the enzymatic oxidation. And there are a lot of reports where people have reported the formation of dityrosine under different conditions, and which resulted into different kind of morphology, and especially there is growth of nanofibers and photothermal film, and also there are some microspheres has been reported. But these are not are the quasar weights. So why quasar weights and then membrane formation? Actually, quasar weights don't have any membrane outside. Might be you are thinking, because quacervates don't have any membrane, how they can hold everything inside? But quacervates, in our case, because it is made by the hydrophobic regions of FFSSFF, and we believe they can hold small molecules and genetic material due to hydrophobic interactions. But because we know living cells need very small solutes. For example, they need salts and other small molecules, even ions, but quacervates cannot hold these small ions and they can escape back to the environment. So this is one of example where people have used interfacial self-assembly to introduce one polymer to make a membrane outside the quacervates. And also they have used POMs to make the membrane outside the quacervates. But this is again a very complex system, and how we can make a single system, single molecule to make the quacervates and then membrane outside the quacervates. Yes, this is one of quacervates example, which is, I didn't put my chemical structure of that molecule, but that forms these quacervates. And when I use enzymatic material as oxidizing agent, these quacervates can be converted to vesicles. And these vesicles, internal core is still kind of environment like the quacervates has, but it has very nice membrane outside.
That's one interruption, please.
Yeah.
That's a interruption, please. आपकी स्लाइड्स जो है वो नहीं रन कर रही है काइंडली.
ये कहाँ पे स्टक हुई हैं?
फर्स्ट वन पे ही है.
फर्स्ट वन पे ही है. काफी लेट बताया है फिर आपने.
हम अपनी वो टेक्निकल टीम से हेल्प लेते रहे कि शायद यहाँ कोई मसला हो.
अच्छा, मेरे पास वो मूव हो रही थी. एनीवे, मैं दोबारा इसको शेयर कर देता हूँ. अनफॉर्चुनेटली ये अब आपके पास कौन सी स्लाइड आ रही है?
नंबर है कोई स्लाइड? लाइफ प्रॉपर्टीज.
ओके, आई थिंक यहाँ से मैं गुजर गया हूँ वैसे तो चलो मैं इसको ऐसे ही रहने देता हूँ. आई थिंक स्लाइड शो पे, प्रेजेंटेशन मोड पे वो स्टक हो जाती है. ओके,
राइट सर, कैरी ऑन प्लीज.
ओके, yes, I was here. Membrane formation. Why we need to make the membrane outside the quacervates? I explained that this is necessary to hold the small salt ions, which are necessary to function as a quacervates for the protocell formation. So therefore, I designed different molecule which can form also quacervates. Unfortunately, you haven't seen that those quacervates in previous slides. So these quacervates can form the vesicles under oxidation conditions, because I use the tyrosine, one of amino acid as a tyrosine, and tyrosine has ability to oxidize to form the dityrosine. So these dityrosine molecules form the vesicles, and vesicles, these are not only quacervates with membrane, but they have various advantages over the quacervates. For example, these are fluorescent, intrinsically fluorescent in nature, and they can little have flexibility in their nature, and it's very similar to lipid bilayer or liposomes kind of structures, but made by the peptides. So we have also confirmed this by TEM, whether these are quasar weights or the vesicles. Yes. So next we investigated how the membrane is either the membrane is rigid or there is any flexibility, just like similar to the membrane of cells. So we observed that these vesicles can change their position, their morphology, and their size mostly remain constant. And for example, you can see in this video, you can see this vesicle is moving and its membrane has flexibility and fluctuation. So, and also there is pores inside the membrane. So it is very nice system which I am investigating recently, and main purpose is to divide this bigger vesicles system to the two quasar weights or two vesicles. But let's see, because it's very complicated system, if I divide this, might be then this system will be disassembled. So I am trying my best, but still it's not working. And overview of the hallmark for the protocell. So if we put as an overview, we have successfully formed the compartments. We have shown the growth of the quasar weights, but it is very difficult to divide the quasar weights again. Recently, there are some reports which people are using different techniques to divide the quasar weights or protocell. But it's not easy to have all these functions in one system. So information processing, no doubt we can encapsulate or sequester the genetic material, but we don't know either these information can be transferred from one system, one quasar weights to the other, just like the living cells are communicating with each other in the body. Energy transduction is also not clear, and mobility. We have shown that quasar weights are mobile in nature and they can move from one place to other. They are dynamic. So it's still, the research is undergoing. It's very difficult to have all functions or hallmarks in one system. But I hope in the future, someone will have all these functions in one system. Next, it's just overview of my previous work, which I have done in my PhD, especially on the biomedical applications field. As we know, cancer is one of the leading diseases in the world, and there is NIH statistic which says that worldwide cancer deaths would be around 15 million by 2030. This is alarming digit, and we have in many different countries, even in Pakistan, we have surgery, chemotherapy, radiation option to cure the cancer. However, all these techniques have various severe disadvantages. For example, after surgery, there is 100% option that cancer will come again. The tumor will grow again. And there is the option, there is the also collateral damage problem. Chemotherapy, you can get, or the patient can get this treatment for long-term term, because this sometimes it takes 6 months, 1 year to complete the chemotherapy. This is also again have a recurrence problem. One, it is very long term. We don't know that patient can survive in that 6 months or 1 year. And also the recurrence problem mean the cancer can come again after 2 years, 3 years. And radiation, this is somehow good technique considered in especially in South Asia, but radiation can damage your healthy cells, and that patient can have many different problems. For example, they can lose eyesight, etc., and even some other functions of the body. So then what is the option? So option could be the phototherapy, which is light-based, non-invasive techniques. There is no side effect, but still, why different countries, even developed countries, are not adopting phototherapy? Only I can say USA in Wellman Center and Canada and Japan, they are using this technique for the only one type of cancer. But why they are not using for every kind of cancer? The problem is because for phototherapy, we need to have some photosensitizer or light responsive molecules. And these light responsive molecules, dyes, drugs have problem. For example, they are hydrophobic in nature and they are not soluble in water. So if someone wants to treat their cancer by these hydrophobic dyes, which are light responsive, and through phototherapy, there are a lot of problems. For example, when you dissolve these or use these drugs in DMSO or other organic solvent which are toxic in nature, you can use DMSO up to 2%, but still once it is in monomeric form, it can spread all over the body. Mean it is difficult to make it selective. Why? Because it's in monomeric form. They are not assembled. And one natural and good thing from the tumors and cancer cells is that they can capture something which are hydrophobic in nature, but because this is already spread in the body, they cannot capture from whole body. So we need to think about some nanotechnology option. So how we can use the nanotechnology or nanomaterials? One option is we can make these hydrophobic drugs by conjugating with different peptides, different proteins to make them biocompatible and to make the nanomaterial, which will help to make the nanomaterial. Other strategy could be we can rationally design the drug carrier which can carry the drug to the tumor site or the cancer site and release the drug as we need. Mean we can control the release of drug at the tumor site. So in my first project, I made these nanodots from this TPP porphyrin conjugate FF system, which were formed through self-assembly, again non-covalent interactions, but these nanodots were highly packed than the quasar weights. Quasar weights are not densely packed, but these nanodots were very packed through non-covalent interactions, and even it was difficult to release that drug. So therefore, we use photothermal therapy. We injected these nanodots to the mice and then irradiated by light, and we saw very nice and useful results after 14 days that all the tumor was eradicated. And my second project was to design some multi-responsive carriers. This is just similar to FF-SSFF system, but it has long hydrophobic chain, and also it has carboxylic acid at the terminus. So this system can form the nanofibers under physiological conditions, and then we can use the hydrophobic drugs to encapsulate in these nanofibers to make the gel. And because there is disulfide bond in this design, and we know cancer cells and tumor cells have glutathione inside, which is again a reducing agent. And GSH, glutathione, can reduce this molecule to release the drug on the tumor side. Once we injected these nanofibers on the tumor side, and the drug was released with sustain and in a controlled fashion. Strategies for antibacterial activities. In my first postdoc, I was supervising my master student, and we designed some small molecules, small peptides, which we used for the antibacterial activity. But here it is overview how we can use the peptide nanomaterials for anti-cancer, antibacterial activity. We can design different kinds of nanomaterial. It could be nanorods, nanofiber, composite material, and due to 4 different mode of action which works for the antibacterial activity. For example, ROS generation, membrane disruption, gene regulation, oxidative stress. These are the 4 mechanisms which these material can kill the bacteria. So we are using recently in my collaboration with Nanocenter Beijing and one of reputed university from Pakistan, we are investigating these quacervates for anti-cancer and antibacterial applications. And I hope I will present somewhere next these results. So I am heading towards conclusion. This is here at Radboud University. We introduce very novel class of short peptides. Nobody has reported this before. Even nobody has reported self-coacervating short peptides before. And we introduced this new class of peptides, which are very similar to proteins, and they can have a phase separation property. And they are reversible in nature, mean we can make the quacervates and we can again deform, or we can cleave the disulfide bond to just this to make the building block again. Or also it is pH responsive, mean once it is pH 7, then if we decrease the pH, then we can get the building block again. So they are capable to encapsulate genetic material and light harvesting pigments, which makes these quacervates as a protocell metal. And they also act as a microreactor for anabolic organic reactions to just mimic metabolic reactions which are happening inside the cells. And I hope these short peptides will have very promising anti-cancer and antibacterial activities and properties. And at the end, I would like to thank or acknowledge my promoters, Evan and Wilhelm, and all the members of Physical Organic Chemistry Department at IMM, Institute for Molecules and Materials. And this is our small group. And lastly, and importantly, I would like to thank my financial support from European Foundation, Marie Curie Foundation. Yeah, thank you very much for your invitation and listening this talk. But unfortunately, I think there was a problem. Even you couldn't see all the slides. That is not good. But I am happy to answer your questions.
So, we are much proud of you that you are one of the forerunners in this field of biomedicine, and I think that you are proud of our nation. Inshallah, we will remain in contact in future. Currently, in question and answer session, I would like to place a question which is related to the NMR.
Yes.
Actually, you have told that in checking the conservates micro reactivity, You work on the principles of NMR. Would you like to explain how the NMR is helpful in predicting the microreactivity of conservators?
Yes, thank you very much. It is very nice question. For example, building block investigation. For example, these are the NMR of these building blocks, mean these are the correct molecules. First, we have done that one. So microreactors, how we can do the NMR for its capability to as a macroreactor. For example, I took 1 milliliter droplets or the quasar weights in NMR tubes. I added two components, which are the reactants of that reaction, mean aldehyde and ketone, inside the quasar weights, inside the NMR tube, and then I checked by NMR to see or track the appearance of new peaks which are related to the product. So after tracking for 48 hours, I come up with the integral intensity of those integral intensity of the protons of that product. So then I apply a model to see the formation of product.
Thank you, sir. One more question. It relates to the history of conservators. In the start, you told that it was initially started by Russian scientists in 1929 almost, and then it was again brought in limelight around 2010. There is a vast gap. Once it was left, then it was taken again. How you can summarize the similarities between the models assumed at that time of 1929, and now it is the era of nanotechnology in 2010. It is again taken in limelight. Were there any similarities in these two models?
Yes, it is. There is one class of scientists which are working especially on the origin of life. Might be I didn't talk about too much about the origin of life due to certain reason. Because that Russian physicist, and before Russian physicist, there was a scientist. I think most of physicists knows. Miller and Corey, which has different experiments also, I think, in the physics. Some people still in the physics books, we can see them. Actually, that scientists were figuring it out that how these small molecules, for example, methane, oxygen, water, these can mix together to form a life. So accidentally, they come up with this small droplet, and they named it as a quasar waves at that time. And he proposed, like, might be these quasar waves would be the best option to study the origin of life. So that was his theory. And then in 2030, Dutch scientist, he used different precursors. For example, he used lipids to make the same kind of quasar waves. And he also supported the idea of that Russian scientist that yes, we can study the origin of life by using the quasar waves. Yeah, but unfortunately, then might be various research towards different fields between these 60, 70 years and 80 years about. And then it was forgotten, and people were thinking only towards RNA world. There are two different kinds of school of thoughts. One school of thought is convinced that might be origin of life was started due to the RNA, presence of RNA. But other is still on the formation of new quasar weights or the new protocell model. And thus now again this second school of thought is emerging and more convincing as compared to the RNA world. Yeah.
Thank you. Audience, please place your questions. We have 10 more minutes, and in this time, your questions will be answered. Inshallah. Any question from audience, please? Okay, sir. One more question from my side. You told that recurrence of cancer can be halted by conservatives, means by the way of phototherapy. I think.
Yes. Yeah.
A brief summary about this can be.
Yeah. Thank you. This is actually very nice direction for the biomedical application, especially I would say there is one group in MIT who is especially working on this direction to use the quasar weights for drug delivery. So what are the advantages of quasar weights as a material? Because as I earlier described, these are not well packed. Building blocks are there are certain gaps between the packing of building blocks. So one advantage is that these quasar weights can uptake or hold bigger quantity. Our payload capacity is more as compared to the nanomaterial. For example, if nanomaterial can carry 1 milligram, for example, 1 milligram drug towards the cancer site or cancer tumor, quasar weights can take 5 milligram drug to the cancer site to increase its efficiency overall. And other advantage of our system could be because still we are using disulfide bond in the center, and we know GSH, glutathione in the tumor and the cancer cell is quite high in ratio as compared to the normal cells. So once our quasar weights will be there on the tumor site, that GSH will release or cleave this building block to release the drug. And other advantage could be because we know the cancer tumors, cancer cells have acidic pH. The pH is around 4 to 5, and we know our system is very responsive towards pH. Once the quasar weights will be there, they will cleave by GSH and they will just become a monomer, building block, by decreasing the pH. Because that pH, we will not introduce any pH. That pH will come from only the environment of tumor itself. So both releasing mechanism will come from the tumor cancer cells actually itself. That would be our that are the advantages of our system for the anti-cancer activities or anti-cancer treatment for phototherapy. Of course, we will use light responsive drugs for the release and for the treatment. Yes.
So much thanks.
I think. Yeah. Someone had a question.
सर बहुत शुक्रिया, थैंक यू. ऑडियंस, अगर कोई सवाल है तो पूछें भाई. स्टूडेंट्स.
One question is there. I can answer that. How you check antibacterial properties? Yes, we checked these antibacterial properties of these quasar weights just by incubating these quasar weights with different strains of bacteria. However, the activity was quite low, but we are trying to make it more antibacterial agent stronger in its efficiency. We are trying different techniques, for example, to encapsulate any metal ions or metal nanoparticle inside the preservates and then used for antibacterial properties. And I am not currently working on antibacterial field, but my colleagues are working in Quaid-e-Azam University and Nano Center. So hopefully we will have nice results in future.
So much thanks, so much thanks. That's all. बहुत शुक्रिया आपका, आपने बहुत टाइम दिया हमें, और आपकी जो प्रेजेंटेशन थी वो मोर देन सफिशिएंट थी हमारे नॉलेज से। तो इंशाल्लाह आपसे इन टच रहेंगे आगे भी। बहुत शुक्रिया।
No problem. Always welcome, and I am very open to talk, to work, and to collaborate with anybody who are interested. And yeah, thank you very much, and thank you Abid and Dr. Bilal and all audience, and of course Dr. Saifur Rahman and Vice Chancellor Sahab. I think that's it.
Thank you.
Yeah. Bye bye. Thank you. Allah Hafiz.